Review of Chapter 17. The title of Chapter 17 is From Gene to Protein. Now, this involves gene expression, or how genes code for traits. Now, the first diagram to look at is Figure 17.3. It shows you an overview of gene expression. Now, gene expression involves a two-step process. The first step is called transcription. The second step is called translation. With transcription, a gene, a sequence of DNA, is used as a template to make a messenger RNA molecule. In other words, you're making a copy of the gene from DNA into RNA. Translation is taking that messenger RNA molecule and using it as a template to make a protein. Now, transcription is called transcription because, like any usage of the word of transcription, the language is being copied, but the language hasn't changed. You're going from nucleotide sequence to nucleotide sequence. Just like if you're transcribing notes, you're not changing the language. Translation is changing the language. So the language is changing from nucleotide sequence to amino acid sequence, because that's what proteins are made of. Transcription occurs in the nucleus of the cell. Translation occurs at the ribosomes of the cell. That was Roman numeral 1. Roman numeral 2 discusses the genetic code. Now, the genetic code is shown in Figure 17.5. Now, this is how translation gets translated. That is how you go from the information from messenger RNA to amino acid sequence. Keep in mind, RNA is single-stranded. RNA has uracil, U, instead of thymine, T. That's the difference. So if you look on Figure 17.5, you're going to see a lot of A's and C's and G's and U's, and some amino acid abbreviations. Now, there are 20 kinds of amino acids, but only 4 are nucleotides. So there can't be a one-to-one correspondence between nucleotides and amino acids. That won't work. There's not enough information there. You can't have 2 nucleotides code for amino acid either, because there's not enough information. There's not enough 2-letter sequences to give you 20 amino acids. So really it's 3. So the magic number is 3. 3 nucleotides code for 1 amino acid. Now, this 3 nucleotide sequence is called a codon, C-O-D-O-N. Okay, a codon. Now, if I were to give you an example of how this works, let's say this is DNA. Now remember, DNA is double-stranded, but only 1 strand gets used. It's called the reading frame. Now, if we're going to transcribe this, transcription, here's our messenger RNA molecule. Remember the base pairing rule? Everything operates on the base pairing rule, but RNA does not have the letter T, so a U goes here. So this will be U, 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 G, G, C, 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 G. Okay? Now, here's a codon, here's a codon, and here's a codon. If you look at the genetic code, you'll see that UUU codes for the amino acid phenylalanine, abbreviated phe. GGC codes for glycine, and CCG codes for arginine. And I do not expect you to memorize the codons coding for the amino acids, but you should be able to use the genetic code or look at the chart in order to know what this is. So in other words, here is enough genetic information to code for 3 amino acids. Now, if you look at the genetic code, you'll also see AUG showing green. That is the start codon. See, translation is like a computer program. It has to have a start command, and that's it. But you'll also see 3 stop codons, UAA, UAG, and UGA. Now, stop codons are the only ones that do not code for amino acids. All the others do, even the start codon. So stop codons are the stop commands in this program. Now, this code has redundancy. There are more codons than there are amino acids. Okay? You'll see that UUU codes for phenylalanine. Well, so does UUC. So with redundancy, more than one codon can code for the same amino acid. Okay? But there's no ambiguity in the genetic code, meaning that a codon does what it does and it does nothing else. UUU only codes for phenylalanine. It doesn't do anything else. But the best part about the genetic code is that it is universal. This isn't just the genetic code for humans. This is the genetic code. The way that this gets translated occurs the same in almost every living thing with almost no variability at all. This is what makes genetic engineering possible. You can take a human gene, say the human gene that codes for insulin, put it in a bacterium, and because the genetic code is the same, it can be transcribed and translated, and you'll have human insulin, just as good as yours and mine. In fact, that's how human insulin is produced pharmaceutically. This is what makes genetic engineering possible. Now, I'll stop this video and I'll continue in part 2.